Assalamu alaikum and good afternoon. Thank you, Katie, for the introduction. So, as I already know, my name is Saad and I'm here to defend my thesis entitled Wastewater Treatment by Microalgae Strains Isolated from an Urban Effluent and Alternative Towards a Circular Bioeconomy. This thesis was a part of Green Treat project currently running at CCMAR, University of Algarve. Research was carried out under the supervision of Professor Luisa Pereira, along with Dr. Etienne Morais as co-supervisor. So I will start with introduction, followed by literature review and objectives. Then I will go through methodology and I will discuss results along with conclusions. Let's have a look at a little bit introduction. So traditional handling of wastewater is unable to remove the emerging pollutants from the wastewater. Usually for the treatment of wastewater, primary, secondary and tertiary treatment methods are used. Global pharmaceutical industry has grown rapidly as well as it has caused a lot of environmental pollution. Due to the current pandemic, the use of antidepressants have been increased throughout the world. Sertraline is one of the very commonly used antidepressant. It is used to treat anxiety, panic attacks, obsessive compulsive disorder, and post-traumatic stress disorders. Sertraline selective serotonin reuptake inhibitors are the most common antidepressants which are a threat to global aquatic environment as surface water systems become affected by wastewater treatment plants all over the world. The sertraline has been detected in the wastewaters of Asia, Europe, and America. However, the concentration of sertraline effluent in the effluent is in European effluent is 30 nanogram per liter after the wastewater treatment. When exposed to sertraline within first 48 hours and the concentration between 4 to 43 nanogram per liter, microalgae has been reported to be immobilized and change of swimming behavior has been observed in crustacea. When exposed for days and concentration more than 50 nanogram per liter, decreased growth and reproduction is observed in water flea and zebrafish exhibit mortality of the embryos. Microalgae are the primary producers in the food chain. There are approximately 45,000 to 100,000 species of microalgae on the earth. Chlorella and spirulina are the most commercially produced and used microalgal species. Microalgal biomass can be used in cosmetics, pharmaceutical, food, and feed industries, as well as for the production of biofuels. Microalgae has also been used for the remediation of wastewater. Algal bloom developed from a combination of different environmental conditions like nutrient, temperature, pH, and hydrology. Having grown in the same environment, they are acclimatized to a variety of pollutants and they are resistant to the pollutants as well. So therefore, they can be used for bioremediation as well as an energy source. When pollutants come in contact with microalgae on the surface of microalgae, bioadsorption occurs on the cell wall, and intracellular biodegradation and intercellular biodegradation occurs with the help of enzymes, and biodegraded products are formed. And pollutants can also be bioaccumulated inside the cells of the microalgae. On the other side, some of the non-biological techniques for the removal of pollutants are hydrolysis, volatilization, and direct photolysis. Previously, Chlamydomonas mexicana has been used to remove 56% ciprofloxacin. Chlorella vulgaris and Microcystis aeruginosa showed inhibited growth in the presence of when exposed to sertraline hydrochloride, and Chlamydomonas acidophila was able to remove 65 to 93% erythromycin. To the best of our knowledge, nothing has been reported using natural microalgal blooms yet. Taking in account the harmful effects of sertraline and the studies that have already been carried out, the main objective of this study was the removal of sertraline using natural microalgal bloom. And the specific objectives were determination of cytotoxicity of sertraline, assessment of sertraline removal potential of microalgal bloom, potential applications of biomass under circular bioeconomy, and lastly, isolation and identification of sertraline-resistant microalgal species. In order to start the experiments, the wastewater sample was collected from Quinta to Lago wastewater treatment plant in Algarve, Portugal. They were brought to CCMAR lab, Biotech lab at CCMAR, and in 1 liter flasks with 50% BG11 and under controlled condition, microalgal production was achieved in 10 days. For the toxicity experiment, 4 different concentrations of sertraline were used in the glass tubes at 25 degrees centigrade with 12-hour light dark period and 100% BG11. This picture was taken on the first day, and this picture was taken after the time interval of 24 hours. In the bubble column photobioreactors, microalgal inoculum was used with 5 different concentrations of sertraline and 100% BG11 in the light and air conditions I mentioned for the sertraline removal experiment. Initial concentration of the inoculum was 0.3 gram per liter, and the concentrations of sertraline were 0.5, 0.1, 0.5, 1, 5, 10, and 10 milligram per liter. Samples were taken after every 24 hours for optical density and pH. This is the standard curve made between dry biomass and absorbance, and which was used for further experiments. And for sertraline removal, the sample was collected at the mentioned hour. pH was maintained at 3 and was frozen for further analysis. For the analysis of sertraline from the frozen sample, solid phase extraction method was used following the following procedures. Mentioned procedure. 50 milliliter of sample was loaded at the rate of flow of 3 milliliter per minute in the elute bound cartridges from Agilent Technologies in the vacuum manifold for solid phase extraction. Finally, the sample was eluted using 1 milliliter of methanol, and the eluted sample was completely dried in nitrogen. For derivatization for GCMS, MSTFA solution was used in the mentioned conditions. This is the gas chromatography mass spectrometry at CCMAR University of Algarve. These are the conditions for GCMS analysis. Initially, the temperature was between 90 degrees for 1 minute, and then it was raised to 180 degrees for 10 minutes, then finally at 300 degrees, which is also the temperature of the injection. And the injection volume used was 1 microliter, and carrier gas was helium. The spectra was obtained at full scan mode along with other mentioned conditions. This is the standard curve for the analysis of sertraline made at GCMS. Limit of detection and limit of quantification is mentioned here. This is the chromatogram from GCMS analysis. This on the top, the red one is total ion chromatogram, which shows the concentrations with peaks of everything that is in the sample. This might also include the noises which also comes along. So we run the sample at 274, which is characteristic ion for sertraline, and in the selected ion mode same, so that it gives the peak of only for the sertraline, and then we integrate and we calculate the area. We have relative intensity on the y-axis and retention time on the x-axis. This is the mass spectrum of sertraline, and here we can see that parent peak of sertraline was observed at 274. This is also this graph is between relative intensity and mass to charge ratio on x-axis. For the biomass analysis, the samples were centrifuged, freeze-dried, homogenized, and they were analyzed for moisture, ashes, carbohydrates, proteins, lipids, and statistically analyzed using ANOVA Tucky test. For the isolation and identification of sertraline-resistant microalgal strains, they were first observed at microscope, and then they were isolated and purified on agar-agar plates with BG11. And finally, the strain identification, for which DNA extraction has been done, and we still have to carry out sequencing. For the first group of results, I will discuss the cytotoxicity experiments. As it can be seen that when microalgal bloom was exposed to different concentrations of sertraline, they could not survive 100 and 1000 milligram per liter within 24 hours. However, the increased growth was observed for other 1 milligram per liter and 10 milligram per liter. Previously, it has been reported single microalgal species have been reported to survive up till 200 microgram per liter. However, in our case, we were able to achieve the results up till 10 milligram per liter. This is the graph which shows microalgal growth of the sertraline removal experiment. And it can be seen that after the scale up, the growth of 10 milligram per liter was reduced, but it did not die for the period of 10 days. It showed it was still alive. And for the others, the continuous growth was observed. Finally, we discussed the results of sertraline removal of 5 and 10 milligram per liter. So the red lines is the rate of removal and blue is the biomass. 5 milligram per liter to remove 92.4% sertraline in 48 hours, and for 10 milligram per liter 91.4% in 48 hours. So it can be seen that minimum concentration of 0.3 gram per liter is required to remove sertraline. At this stage, we can also hypothesize that bioadsorption occurs where sertraline accumulates in the dead microalgal cells. In order to test this hypothesis, we are currently running these experiments with inactivated microalgal cells. This is the overall graph of biomass evaluation. For all the 5 concentrations of sertraline, we see there is no significant difference was observed for proteins and carbohydrates, and which was also confirmed with ANOVA Tucky test. Since we have large quantities of proteins, one of the proposed use and of carbohydrates, one of the proposed use can be biogas or bio-oil production. Here is how circular bioeconomy works for a sustainable future. In order to sertraline contaminated wastewater is treated with microalgal biomass, and that biomass is used for the production of biofuels, which then produces energy, and that can be used to treat the water. And on the other hand, the treated water, sertraline-free water, can be reused or safely disposed according to the standards regulations. These are few of the sertraline-resistant strains which were observed on the microscope. The strains were then grown on the agar-agar plus BG11 plates and observed on the microscope. This one is assumed to be chlorophyte on the basis of morphology, and this one is assumed to be cyanobacteria, and the scale is 10 micrometer. All this allows us to conclude that for the cytotoxicity analysis, we were able to achieve 10 milligram per liter growth of natural bloom, but 100 milligram and 1000 milligram per liter could not survive. And the sertraline removal potential of the bloom, they were able to remove 92.4% and 91.4% of 5 and 10 milligram per liter sertraline respectively in 48 hours. Some of the potential uses of the biomass could be in the biogas, bio-oil, and bioethanol. Isolation of sertraline-resistant microalgal species, they are assumed to be chlorophytes and cyanobacteria. However, we still have to confirm the identification by PCR. In the end, I would like to thank my supervising committee for their help throughout the research, Marbiotech Lab for their support and coordination, and IMBSC Coordination Office, and last but not the least, my friends and family for the help throughout. These are few of the references that I consulted for this presentation. I will be happy to answer any questions.